Only mode.
Hello friends, it's Joe with my friend Philip Norris MD, and I need to start with the great irony of your name, which is one letter away from the most toxic company out there. I used to work for Philip Morris.
Oh, really?
Yeah. Well, indirectly, I worked for Kraft Foods, which was then owned by Philip Morris.
Yeah.
And so to have Philip Norris MD, it's almost like an oxymoron just meeting you.
Yeah, so when I was training, I did full-time clinical work, and I would see people with lung cancer. I was in North Carolina at the time, and they looked at my name and they said, "Really?" No, it's Philip Norris, one letter off.
Mm-hmm. Well, despite your unfortunate nomenclature, I understand you're a very smart man, and there's a certificate on the wall there. What does that say?
Oh gosh, I don't know which one we're looking at.
Right over your left shoulder there.
Over my left shoulder. No, you think my right shoulder. So what you're looking at,
man. It's right there on the wall.
Well, there are a bunch of certificates, so you can't really see them all. I have all my degrees. I wear my
Your degrees on your sleeve.
The thing right behind me is a pathway of T-cell activations. Those are all little molecules that interact together. So my lab is involved with basic immunology research. Started off cloning T-cells and HIV infection back when I was in Boston, and then came out here and have really focused on how the body's immune system responds to viral infections is the main theme going through the laboratory. And I'm based at a blood bank now, so we look at slightly different sets of people than the usual hospital doctor, which has its pluses and minuses, and we'll probably talk about that today.
I'm sure we will. And when you made the adjustment, you made it a little worse. Bring it down just a little bit because there's the overhang of your...
I was trying to show you my certificate.
Now,
is that better, worse?
No,
oh, heck.
Yeah,
sorry, I'll keep playing with it. All right. Go for it.
We'll correct you in post, as they say. Oh, actually, that's the worst of all you've done.
Why is it so bad? Is it the lighting?
No, it's the thing, whatever it's right above your monitor. If it's like a credenza or something, it's there. Oh, wow. Ladies, take your screenshots now.
Too much, Joe.
Okay, so when it came time to give you like a bio line as I wrote this up, I didn't know which of the 4 things under your title I should give. So tell us the 4 things that you are professionally, and then we'll figure out, you'll get the nod of, wow, this guy knows what he's talking about, and then we'll listen to your great wisdom.
Yeah, well, the titles are probably better than the actual person. So I work for Vitalant, which is a national blood bank. We're the second biggest after the Red Cross. And I'm vice president. Yes? So I'm vice president of research at Vitalant. And within Vitalant Research Institute, I'm director of laboratory sciences. I'm not sure exactly what that means, but I run the basic science part of the laboratories, and I'm a senior investigator at Vitalant. And then we're located in San Francisco, so all of our faculty are also on faculty at UCSF, University of California, San Francisco, at the medical school. So I'm a professor of medicine and laboratory medicine over there. And so as the blood bank, our home department is really laboratory medicine that does testing and runs blood banks. And I also was trained as an infectious disease doctor and still see patients at San Francisco General Hospital through the Department of Medicine. So about 1 month a year, I'll see patients. And actually, I was on service at the end of January and was convinced that I had COVID. Not at the time. I was sick. I had started off with a diarrheal illness and then got a terrible viremia and felt terrible, stayed in bed, and then developed a cough for a couple of weeks. Certainly consistent with it. I have had an antibody test because we have some of the earliest antibody tests here, and I was negative. But as we'll hear, I still could have had it. So I think we'll go through some of these mysteries as we chat today.
In fact, next week, sight unseen, I invited you back for a second one because next week you're going to maybe have a friend with you, and we're going to talk about how to test people before they go back to work.
Oh, interesting. Okay. Yeah, I'd be happy to come back in a week.
You sound surprised. I thought we agreed on this. Well, you're busy next Tuesday at this time.
That sounds good.
How many? Is it a dozen? Is it hundreds of studies are going on right now to figure out this virus and how to defeat it?
Oh, I would say it's thousands. So basically, every dummy with a pipette, which describes all scientists, have gone from whatever they were doing to something COVID related. Now, I couldn't say everybody, but really, it's been impressive how the whole field, and I'm sure the people on the call listening feel the same, that this is a huge crisis, and everyone who has the ability has tried to respond. So there are literally thousands of studies going on across the world.
Maybe you'll touch upon some of them that are most promising, and if a visual aid will help, if you want the screen, just let me know, or we can just talk, that's fine.
Yeah, I wasn't sure exactly what you wanted, so I have, I have a presentation that we gave to CEPI, the vaccine initiative in Norway.
I'll give you the screen because I suspect that what you'll share will be completely new to many of us, myself included.
Yeah, so it's pretty hardcore science. Is that what you want, or do you want more of a high-level discussion? Let's
give it a shot, and if it's too much, my audience can write in questions like, make it stop, or this is great.
Good, okay, I won't be insulted. I won't be insulted at all. Okay, so I'm going to show my screen then. You're giving me an invitation.
I did. That's your email.
Now, how do I get my slides to show if I...
There on the bottom, it says CEP Norris SIM, and it says USFF. Yeah, click on that.
Do I need to bring...
just click on the PowerPoint that's relevant.
Can you see the PowerPoint now?
I can.
Okay, so I'll put it in presenter mode. It says Understanding COVID-19.
Brilliant.
Okay, good. All right, so I will run through. I have about 20-something slides here, and if it gets too boring, go ahead and stop me. But
this is the audience participation part, so please liberally leave comments and questions, compliment us, whatever you like. Go ahead.
Okay, so I'll just run through this. This is something that we at the Blood Center think are some of the pressing questions for the COVID-19 outbreak. So the innate immune response is what happens earliest in an infection. So the immune system recognizes danger signals. They're not specific to any given virus, but all bacteria and viruses sort of have this common theme of inflammation that they might induce. So that's the innate immune response. Doesn't need any training, you're born with it, and it gets off and running. It releases cytokines, or the things that make you feel terrible when you have a fever. That's not the virus, that's actually your cytokines flowing around your body that have been triggered by the innate immune response. So does that timing and how vigorous it is have an effect on outcome from COVID-19? And we think it does. And then this is really interesting. The second bullet point, we'll get into this. How common is an ineffective immune response after natural infection? And I'll talk a little about that. And does the region of the virus targeted by B and T cell responses associate with disease outcomes? So B cells make the antibodies, and then T cells either help the B cells or they can kill virus-infected cells. So does that have anything to do with disease outcome? And then this is really interesting, the last point. We don't really have a lot of data on this, but do pre-existing cross-reactive coronavirus responses predispose to severe disease? So as many people probably know, the common cold, about 30% of them are caused by coronaviruses. There are 4 circulating seasonal coronaviruses. And what's really interesting about those viruses is they don't mutate much. So some viruses like influenza mutate each year and you have to get a new vaccine each year because the virus changes. Well, the coronaviruses, they don't change as much, and you'll get infected with the exact same virus 1 or 2 years after you've had it before. So you constantly get reinfected with these coronaviruses, and that's where that kind of gets to that second bullet point where I said an ineffective immune response. So these seasonal coronaviruses don't do a great job eliciting antibody responses. So that's going to be really important as we think about how to vaccinate against disease, and also if you get infected, will you be protected in the following seasons? So this is a schematic that I made, and it kind of gets to what I think happens in the immune response to coronaviruses. So in the first bullet here, you can see that maybe there are data, animal data, to suggest that if you have a delayed interferon response. So that's the cytokine that's produced by your innate immune system. These are macrophages here. They see the virus, and then they produce these little dots or cytokines that then activate other cells like dendritic cells or neutrophils on the right here. And if it's delayed, you might have a dysfunctional B and T cell response down the line. The other thing that happens is there's inflammation in the lungs. We do know that. So part of what happens is not just virus getting into your lung cells and killing them off, but actually the immune response. This is a neutrophil here that's spewing out all these toxic molecules. So we know that tissue damage happens when an overexuberant immune response happens in the lungs. So why is that? Is that due to bad triggering of the innate immune response? We don't know. Another interesting thing is that clinically, when someone gets COVID-19, they get sick, they may have a fever, and then about a week into it, the fever may resolve or not, but antibody responses come up, and that's what you can see down here in number 3, the B cell, or 4, the B cells are producing antibodies. But that is the point when the lungs fill up with fluid and things really turn south. So people need increasing oxygen requirement, they may get intubated, they may die of lung failure, and it all happens around the time that the T and B cell responses are kicking in. So that's a really interesting puzzle. Why is it that when the antibody response comes up, the lungs head south? So that's another area that needs to be researched. So another thing that we notice clinically with COVID-19 is that people develop a T cell lymphopenia. So if you look at their blood counts, you'll notice that there are more neutrophils and fewer T cells. And so we do know that the virus can infect T cells at least in other coronaviruses like SARS, the first version. So one hypothesis is that the virus can induce the T cells to undergo programmed cell death, or what we call apoptosis. So the virus interacts with the T cell, may get inside it. We don't think it can replicate actively and make new viruses in the T cells, but it can wipe them out. So maybe it's inducing an immune defect early on, and that may explain why we don't make very good antibody responses in some individuals. And we think that weak T cell response may impair the ability of the T cell to help the B cells make antibodies, because that's what happens. The B cell recognizes something is foreign, and then the T cell will come along and help it and make strong antibody responses. And then the other question we have, there's a theory that we know that older people do worse with coronavirus. So most kids, you've probably heard about some of the autoimmune things that are going on in kids, but in general, kids do very well with coronavirus infection. So why is that? Well, maybe because they don't have strong responses against the seasonal coronaviruses, and older people, maybe they have their immune system is imprinted with these older coronaviruses, the seasonal coronavirus, and that cross-reactivity, maybe that's inducing a response that's not protective and actually makes things worse. So that can happen. There's something called antibody-dependent enhancement, where the antibody can actually, if it doesn't neutralize the virus and bind to it and stick to the virus and neutralize it, it can actually help the virus get into cells. So that could be happening. So that's another theory that we want to explore. Does your immune state when you go into the infection determine the outcome? So that's sort of a broad level of the immune things we want to look at.
Before you move on, go back to the previous slide, just a sec.
Yeah.
This is a public service announcement for everyone on the call. I didn't understand most of what you said. It sounded impressive, but that's okay. That's just me. I want to give the audience a chance to tell me kind of where they are relative to what you shared. I think the bigger point here, as is the case with most of these calls, I know a smart guy who knows a lot about this. So when you have a question about this kind of thing, you can ask me, and I can find Dr. Norris, and with his generous time, we may be able to get you closer to what you need so that we can do something about this pandemic. So I listened with interest, and I can go back and play it really slowly and try to track things, but My friend Fred writes, a quick or brief description of B, T, macrophage, and neutrophil might help much of the audience, and I know I'm included in that description. But I have others who are like, excellent, I understand, keep going, that kind of thing. So thank you for the feedback, folks.
Okay, well, let's talk a little about the immune system. I glossed over it. So you have cells that are part of the innate system. So these cells run around and patrol all the time. And if you get a bacterium in you, like you cut your finger and some bacteria get in there, this macrophage is going to come along, and you can see it's just a blob-like thing here. And here it's activated on the right. I hope you can see my cursor. Mm-hmm. So you can see I had all these projections out. And what it will do is eat invaders, and then it'll actually chop up the invaders and display little bits of it on its surface so that it can be recognized by these T and B cells. It also will, another cell that's similar to the macrophage is a dendritic cell. It has these big, long projections sticking out, and it sits in tissues, and it picks up antigens, which is foreign proteins. So it picks up stuff that it finds, chops it up, displays it on its surface, and then it will show it to T cells and B cells. Now, you guys all know what a lymph node is. You get a swollen lymph node in your neck when you have a sore throat. So the lymph node is where all these cells get together, the dendritic cells, the T cells, and the B cells, and they have a party in there. And so you're born with T cells and B cells with billions of different combinations of their receptor. So this is a T cell receptor here, and basically you may have a T cell floating around in you that recognizes SARS-CoV-2, the virus that causes COVID-19. And unless you get infected, that T cell is just going to wander through life forever. These things can last your lifetime, and it will never find its love match. It will never do anything. But if it then runs into SARS-CoV-2 because you have it, and it sees a cell that is infected with it, and the protein, the little short piece of protein that it recognizes is on the surface of that cell, it will go nuts. It will divide. It will make billions of itself. It will really, it's exponential growth because they'll all double, double, double. And so within a week or two after infection, you will have T cells and B cells that have undergone this remarkable expansion. And the T cells, there are two types. There are Cd4 T-Cells. These are the ones that are infected by HIV. And they're kind of the generals of the immune system. They help the B cells make antibody, and they can help CD8 T-Cells do their thing. So they run around and coordinate these other cells. And T-cells, they're drawn as these little round things, but they actually are crawling all over the other cells all the time, surveilling the immune system. They also knock out cancer cells. So they don't just kill virus-infected cells, but they'll get rid of your cancer cells as well. They're pretty useful. And then the CD8 T-cells is the second main type of T-cell. And what they tend to do is they look over all of your cells. All of your cells are recognized by CD8 T-cells. And they'll look, when your cell is making proteins, which it always is, it chops up a little bit of everything it's making and displays it on the surface of the cell. So if you have a skin cell and it's, say it's infected with herpes virus, there will be herpes virus proteins displayed on the surface of the cell. And if a CD8 T cell comes along that recognizes herpes virus, it will kill that infected cell because it says, hey, you shouldn't have that protein on your surface. So that's what the T cells do. And then the B cells, they talk to the T cells, but what they do is they make antibodies. And these are these little Y-shaped proteins here. And the business end of the antibody is the top of the Y. And that binds with exquisite sensitivity and specificity to proteins. And so if you're a SARS-CoV-2 specific antibody, you will recognize a portion of generally the outer part of the virus, so the spike protein, and the nuclear protein is also recognized, but let's focus on spike. So this B cell will recognize the spike protein, and then when it gets turned on, it will make billions of copies of itself, and then it will flood the bloodstream with antibodies and surfaces like mucosal surfaces like the lung. There are antibodies that specifically go to the lung. They're called IgA, immunoglobulin A, and then immunoglobulin G runs around in your bloodstream. So these will bind to the virus and hopefully neutralize it, knock it out. So if you get coated, the virus gets coated with antibodies, it gets knocked out. And then up here in the another part of the innate immune system is what's called a neutrophil. And this is loaded with toxic granules. You can see it spewing them out here. So this cell goes around, and if it sees a cell it doesn't like, or a bacteria it doesn't like, or a parasite, it will unload its payload of these toxic granules and kill the cells around it. It's like a little bit like a bomb going off, though, and that's what happens in the lungs with a pneumonia, is you have all this inflammation that really blocks the ability of the lung to work because it fills up with fluid. So that is sort of a immune 101 as to what's going on.
Very appreciated, thank you.
Sure. So if we go to the way we do research, which is a little different from the usual hospital-based researchers, here we have a situation where we have a bunch of healthy people, and then maybe one of them has COVID-19, right? So one of the things we're looking at is we test blood donors using a nucleic acid test. So here come our test manufacturers. And we work really closely with the large manufacturers for nucleic acid testing. I think Roche and Grifols are the two biggest in the blood bank space. Grifols is pretty close to Hologic. They just split recently. And Hologic tests outside the blood bank space, Grifols tests within it. So we're looking to see in our blood donors, if they come in and donate, maybe they're incubating the virus, they don't feel sick yet because you can't have a fever and donate blood, but what if they have nucleic acid running around in their bloodstream? So we've seen this. We test about 1 million units of blood. Well, we test about 75% of the nation's blood supply through our testing lab in collaboration with the Red Cross and One Blood in Florida. So we test a whole bunch of people, millions of people every year, and we can pick up things like West Nile virus. We'll pick up 50 people a year who have West Nile virus and don't know it. And then we can enroll people into serial follow-up, getting samples. So blood donors are great research subjects. They already have the altruism gene. Mm-hmm. So if you just gave blood and you say, hey, I'd like to get even more blood out of you, they say, hey, no problem, when do you want me to come? So we're hoping to enroll blood donors. This protocol is still being prepared, although it's received funding from the NIH, where we will get them to come back and give serial samples. And what this will allow us to do is look at the earliest immune responses and say, what is it about the early immune response that predicts outcome? Because if we detect someone who's well when they donate blood, they will go on to get sicker, and they may go on to have a very mild infection, or they may go on to have a really severe infection and get hospitalized. So we'll be able to look at the early samples and say what happened. And then later on, at the end of the follow-up, we'll be able to say, well, who really made an antibody response that stuck around? Because one of the things we don't know is how long will antibodies last after we get them? You know, in a seasonal coronavirus infection, they don't last that long, and people get reinfected all the time with the exact same coronavirus. So I suspect we will see that with SARS-CoV-2. The hope is that a repeat infection will be mild. We see that in the common cold, that if you get the same cold again, you tend to have a milder illness. And if that happens with SARS-CoV-2, it could, in a generation, become the 5th seasonal coronavirus. And, you know, kids would get infected with it in childhood, they'd have a mild infection, and then they would go on to have, you know, multiple rounds of this throughout the rest of their life, but it would just be a common cold. So that's a real distinct possibility that that's what's going to happen. Now, that doesn't help all us old geezers who will get sick now, so we need to figure out something to do now. The other thing that we're looking at, which is really fascinating, is convalescent plasma donation. So I think probably most of the people on the call have heard about this. If someone has been infected with SARS-CoV-2, they survive their infection, we have them come back to the blood bank and donate plasma, because that plasma is loaded with those antibodies that we were talking about in the last slide, right? So the B cells have fired up, they've made high levels of these antibodies that can neutralize the virus. But what we're finding, and is really interesting, is this. So this is a neutralizing antibody assay. This was developed by Graham Simmons, one of the scientists at our institute. So I think I have a slide as to how it works later. But basically, you can measure the entry of virus into cells, and that's what we call 100%. And if we add the patient's serum and it has antibodies in it, it will block the entry into the cell. And the more potent the antibody, these are very potent antibodies down here, and then these are not very potent antibodies here, and these antibodies don't do any neutralization up here. So it's still at 100% entry into cells. So what this graph shows is there's about a 100-fold difference in people's ability to make antibodies to the virus that neutralizes the virus. And I'll show you in another slide, about 5 to 10% of people who have survived infection, we had documented infection, they had virus in their nose, we detected it with PCR, they don't make an antibody response that's detectable after infection. So what is going on there? And that's a real research question that we need to answer. And that has huge implications for vaccines as well. If natural infection doesn't even induce an antibody response, how in the world is a vaccine going to induce a strong and protective antibody response? And I think I'll skip this last part of the slide. It's really looking at the innate immune response, these cytokines, early in infection. So we'll do that as well. So here I'm not going to go into this in great detail. This is how we're going to answer those questions I had on the first slide. So we're going to look at the innate immune system. We're going to look at cytokine testing and look at the monocytes, those innate immune cells, and see what's different about them in people who go on to have mild infection versus severe infection. We're going to look at the antibodies. We have a whole bunch of assays that test a lot of antibody functions. So antibodies, I've talked about them coating the virus and neutralizing it, but there are other things that antibodies can do. They can coat the virus and make it more sensitive to killing by other cells. So there are a lot of things that antibodies do, and we'll go into detail about all those different functions. And then my lab will look at the T-cells and what happens with the T-cells in these responses.
I have two things to mention. One is, while you were speaking, I just did a quick search on your company, and the first press release talks about how basically you're imploring people to give blood to help alleviate the shortage. So for those of you on the call who are so inclined, please do give blood. Second is, because many of the folks on the call are in the manufacturing space, I see lots of boxes there and vials and things like that. Are there any things that the industry, your industry, is short of right now? We don't have enough what? We hear about testing, but we don't really understand what those components are and what, if anything, my community can do to help alleviate the situation.
Okay, so Joe, I'm going to go to your first point first. Not only should you donate blood, because June is historically a tough month for us because people start to go on vacation and high schools are out, so we don't have the high school donors, and it's even worse now. So it's really interesting what happened with the blood business. So, you know, we collect blood and we sell it to hospitals, and the first thing that happened was demand went down about 30%, which was really rough, because if you sell blood and we have a lot, the costs are fixed for the most part, it's the people, and suddenly you have 30% less revenue. That is devastating. So we had to have furloughs, and it was a mess. Wow. Now suddenly, yeah, I didn't realize it either, but it was a real problem. Now demand has bounced right back with all the elective surgeries that everybody had put off or getting done, and we're short of blood. So one of the things they're doing, they just started yesterday, is blood donors who come to Vitalant sites, if you come to donate blood, you'll get an antibody test. And we use a fantastically specific antibody test. I'll tell you a little bit more about them. But this is really important, and maybe I'll digress a little bit now. Mm-hmm. You get an antibody test, so come donate blood. It's good for you and good for the country. The thing about antibody tests is you have, in a low prevalence setting, which most places outside of New York City are in the US, the specificity has to be incredibly high, because if you're testing a population where the prevalence is under 1% and you have a 99% specific test, most of your positives are going to be false positives. So there's a paper under review at Nature right now. It's published in medRxiv, which is a website of preprints, so pre-published papers, where we looked at, I think it was about 1,000 patients and had 4 positives on the Abbott assay, which is very specific. It's 99.6% specific, but it's a very low prevalence in San Francisco. These were San Francisco donors. And we tested them on the ortho assay, which so far has had 100% specificity. And of the 4 positive on the Abbott assay, only 1 was positive on the ortho and also in our in-house neutralization assay. So there was 1 true positive and 3 false positives. So that tells you if you're in a low prevalence setting, you really shouldn't be doing antibody testing, particularly at the individual level, to try to return someone to work. And maybe we'll come back to that next week. So that was my diversion on antibodies. And then your second question was really about supply chain. What do we not have? So I think we're still looking at masks, or the personal protective equipment is still short. So not only masks, but there are these positive air pressure respirators. They're little mask-like, they're helmet things that you put on your head. So the disposables for that are backlogged about 6 weeks.
Well, if you would, the scarcity, is it a function of out of stock, out of budget? Because I have folks in the network who are telling me, Joe, I have 5,000, 10,000, 50,000 lots of masks that I can make available. Do you want me to put them in touch with you?
Yeah, that would be great. So I know that and the mask issue is...
Some of them are on this call, so if you want to type in, hey, I got some masks, this is your chance.
Yeah, that would be great. And I can pass that on to our supply chain folks, because I know that some of the things that we buy a lot of, we're able to do because we place large orders for our blood bags and those sorts of things that we're generally ordering. But things that we're ramping up, like masks, we didn't order a whole bunch before. So I know that has been an issue, and we're rationing them to 2 masks a week for our folks here. And I think it's going okay, but I think we still have N95 shortages.
Well, if nothing else comes out of this call that we get you some of the stuff, I'll be more than thrilled. Let me ask you, and this maddens me personally, I don't want to take us too far off, but I had the good fortune of studying at the London School of Economics in 1986, and as a result, I can never give blood again because that was around the time of mad cow. Is that ever going to go away, or am I never going to be able to give blood again in my whole life?
Yeah, really good question. So I also have that mad cow deferral. I do think that will go away. I haven't heard a lot.
You're desperate. I mean, this is silly, but I'll ask it because it's just me. Is there ever a time where you can relax whose blood you'll take for an emergency like this? Like maybe if you just use mine for testing, or I did an apheresis, is there any benefit out of using my blood, or just, sorry, no, I appreciate your good intent, go do something else?
Yeah, I know, really good question. So for the mad cow thing, I think they're not ready to do it. The FDA is very conservative. I think that as time goes on, it's clear that there really isn't a big risk. So I do think that will go away. But in this crisis, so there was a lifetime ban on donation for men who have sex with men. If it was after 1978, you couldn't donate in the US. And then the data, most of it generated out of our blood bank here, actually. One of our researchers really was interested in this question, and basically the data that he generated convinced the FDA to relax it to a 1-year deferral. So if you've had sex with another man within 1 year, you can't donate, but otherwise you can. And then with this crisis, the FDA just unilaterally moved it to 3 months deferral. So they did react to the crisis by decreasing that significantly. And we're monitoring at our place through an active monitoring program to look at risk factors and people who test positive for HIV in our blood donation testing lab. They're amping up that surveillance to accompany the relaxed guidelines. So they did do some relaxation, but not on the mad cow front.
Maybe you could put in an email to someone.
Yeah.
I save all the most ignorant questions for myself. You talked about men with men. Is there anything on women with women, or they're free to do whatever they like?
Yeah, no, you're right. So even there is no, and that's part of the objection of the MSM community is, you could have 100 sexual contacts in the heterosexual world, but you're not deferred, whereas the MSM is. Now, it is a higher risk. We just know there are more infections in that group. So that gets a little bit into the politics, but so we're trying to generate the data here to inform that in a scientific fashion.
Okay. As long as you send in the email about Mad Cow, I'm happy.
Okay, will do. Okay, so I'm not going to go through all the slides in this talk because I think they're too detailed. These are, for those in technology, these are some of the technologies we use. NanoString is a cool machine that can measure both RNA targets and protein targets.
You'll be able to share the slides with me to share with the community?
Yeah, I think I can do that.
Okay, we'll check with the powers if so.
Yeah, let me think, because there are some other people who helped me with this presentation. I'll check.
Okay.
But I'm not sure this is totally useful for your community.
NanoString is just up the block from me here in Seattle.
Oh, interesting. Yeah. So this is the kind of thing you get differential gene expression in different populations. That's what the output looks like from a NanoString. Another thing we do a lot of is single cell RNA Seek. So you get reagents flowing in one channel, single cells coming in another channel. You pop them together in a little oil droplet, and then you can look at all of the RNA that's expressed in that cell. And it's really cool because the data that you get, there's so much data that you have to have bioinformatics to really process it. And so what this is, T-statistic, T-distributed statistic neighbor embedding analysis, TISNE is what everybody calls it. And so you can see these populations. These were different populations of cells that were either latently infected with HIV, meaning it's a quiet infection that's just dormant, versus a productive infection where the cells are spewing out HIV. And you can separate all these cells based on their characteristics. You basically reduce all the complexity down to these 2 dimensions so that you can visualize the data. And you can actually go in and see which genes are different between these populations. So that's what we do with single cell RNA Seek. And we'll be doing that with COVID-19 to see what is it that's different between the people who go on to get sick, really sick, and those who don't. I do want to show a couple of the antibody slides. So this is, not this slide, this one I think is interesting. So these are people who we've detected positive. These are convalescent plasma donors. They know they were infected. And we find that the neutralizing antibody correlates pretty well with the ortho assay that we're running, a signal to cut off here. But what's interesting is there are some with a strong signal to cut off. This is a log scale, so this is 100 here. There are some that have fairly high level binding antibodies that don't neutralize. So the other thing we find is there are quite a few people who are negative. So as I mentioned, about 10% of people who are convalescent plasma donors, or 5 to 10%, don't have detectable antibody responses. So that's another thing that we've found so far. We don't have a whole lot of data to present so far, but we have some. I'm going to skip all of these slides. This is one of the other things we want to do is map the epitopes. This is the virus structure here. I think it's just so pretty. And there's a trimer on the surface. This is actually chikungunya virus, but it's a similar thing we would do with COVID-19 is map the antibodies and where they bind to the virus structure. So that's another thing that we're doing. And by doing that, you can make therapeutics that actually will potentially, you could just give this monoclonal antibody as a therapeutic. And there are people
I'd like to ask our audience, in addition to being pretty, are the things that the doctor is discussing, do they affect your work personally? The kinds of things that he's talking about. You just shut off your camera. There you go.
I got it. I was trying to shut off the slides. How do I stop the slides?
I can make that stop.
Stop showing screen. Okay.
Oh.
So I think we can talk. I mean, I've shown you guys a lot of slides. Let's talk now.
Giuseppe asked the question, what kind of test is reliable to test the presence of COVID-19 in donators? And what tests to check the immunity to subjects who already had the virus?
Yeah, really good question. So it was Giuseppe?
Yes.
Okay, so as Giuseppe knows, there are RT-PCR or transcription-mediated amplification ways to detect the RNA. So you're detecting the presence of the virus. And that is mostly in the upper respiratory tract. So your nasopharynx, they stick that swab and it goes all the way back. I'm going to do it right here. It goes all the way back to here in your nose.
My goodness.
It's a lot of fun. It's a long swab and goes all the way back to the angle of the ear. And then they may twist it around a little bit and then take it out. It's unpleasant.
It sounds
Yeah, my wife just had elective surgery, and everybody getting elective surgery now has to have a test a couple of days before they come in for the surgery to make sure you're not going to contaminate the OR. That test is looking for the presence of the virus itself. And we do sometimes find it in the blood. We're concerned about it for blood safety because we don't want to give a transfusion that has virus in it and make someone sick. Someone who's already sick gets sicker. So there are assays that are very sensitive for that. So Grifols is the platform that we're using here at the blood bank. We have them in our research labs. We actually have the commercial testing platform. So they're making reagents, and we're going to be screening the blood supply for specific research donors. So we're making actually pools of blood samples to be efficient. So with 3,000 pools, we can test 300,000 blood samples. And we've started testing. We haven't found any positives. There was one at Stanford that was very low level. There's the cycle threshold. So every time you do a PCR reaction, it doubles. So at 40 cycles, you're at 2 to the 40th power, which is a lot, right, in terms of expansion. And this sample turned positive at maybe 30 something or 40 cycles. So very low levels detected in one blood donor at Stanford. There have been some reports out of Brazil of people who've recovered giving convalescent plasma, and they found some positives in Sao Paulo in Brazil. That hasn't been published yet. So there are isolated reports of it being in the blood. There was also a paper that recently came out that showed they took people who had the virus in their blood and they tried to infect a cell line, and they couldn't grow the virus in the lab. So it may be there, but not infectious. And that's another thing I want to touch on when you're talking about in the nose. So you've probably heard of these people who are positive in their nose for months after infection. And the question is, are they still infectious? So we found that people can carry the virus in their nose after they feel better, after they're, a very sensitive test will pick it up still, but is it infectious? So there've been a couple of studies, one out of Germany and one out of South Korea, where they've looked at this, and after about 8 days, they can't culture the virus. Now, maybe some very, very low-level virus is there that's infectious, but it looks like once the antibody response kicks in, remember those B cells are making antibodies, once that kicks in, you may still find the nucleic acid from the virus, the RNA, but it doesn't look like it's infectious. So maybe it's just pieces of virus, maybe it's neutralized virus, but that's what we think so far.
Okay. I want to ask, it was 2 weeks ago, my guest was Carla Mann Woods, and she talked about, well, it really devolved into who to believe, basically, because there's a lot of information and potentially misinformation out there. And that was around the time that the president announced that he was taking hydroxychloroquine. And for those of you who were on that call, I want you to congratulate me now for being able to say hydroxychloroquine, because I couldn't do it 3 weeks ago. Anyhow, she brought up the specter of Fauci and his interest and, potential biases. And I said, I lament, I had hoped, thought, perhaps naively, that there is a body out there that is apolitical, that something like the CDC could be not influenced by outside forces, but rather just, we're here to protect the world's population from disease. And I left the call despondent, thinking that no, as long as there are humans involved, there will be personal interests and biases and the like. Among the things that we said we would talk about today were the studies in the field. Do you have a perspective, an update, anything you can share about work being done around hydroxychloroquine?
Yeah, well, I just know what you do from the press, really. So I can tell you the mechanism of action. Early on in this outbreak, in I think January, I did a journal club here on remdesivir and hydroxychloroquine because they were shown in vitro in a model of cells trying to infect them that both could block infection. And the guy in the office behind me, who's much smarter than I, works on viral entry. No, he's behind the bookcase. He's hiding.
Oh, okay.
So the thing about hydroxychloroquine, it works in a couple of ways. So it blocks acidification.
Are you live stream?
Of the hi there.
The world says hi. You please go away.
Well, I just got accepted.
So, but you did?
Where?
To a job.
My son's going to work at Cold Stone Creamery, so the virus is on hold and everything is better. Okay, I love you, son. I really do. Please go away. That's Lucas, everybody. Yay for Lucas. Get out.
Job Lucas. Wow, that's exciting.
Right.
So hydroxychloroquine acidifies the compartment of the cell called an endosome. But basically, it blocks that acidification process so the virus can't get out. So that's one of the mechanisms. But apparently, there are proteases on the surface of the cell that make hydroxychloroquine really, it worked in that cell line, but actually in human lung cells, it doesn't work, according to my lab mate. And the thing that concerns me as a physician is that I know it has cardiac toxicity. So if it has poor therapeutic profile where it may have some minor effect on the virus, but not really much, combined with toxicity, you have to give it in high doses. The data that I've heard is that there are increased deaths in the hydroxychloroquine arm, and the WHO has stopped trials on this. So I do think that the balance between safety and efficacy is not good for hydroxychloroquine in this infection.
One of the things, and I imagine doses is a big part of it, but Carla pointed out that she has, and I likely have, taken hydroxychloroquine when we were going to take a holiday in Africa. I went on a photo safari.
Yeah, much lower dose.
Yeah, so it's not a matter of, if I take a pill, I'm not going to die. But if you take it as a prophylactic for this condition, you're probably taking much more than I did for my vaccination, as it were, or my prophylactic.
Yeah, and I think that particularly for people being treated in the hospital is where they give the really high doses for people with active disease, and that's where the trials have been going on. For prophylaxis, I doubt it does anything at the low dose. If you take it at the malaria dose, I doubt it would do anything in terms of protection.
Just on this, just if you can help me equilibrate. If I take the equivalent of one dose, whatever unit you're talking about, to go to my trip to Africa, what would someone who says, I'm taking hydroxychloroquine to protect me from coronavirus be taking? Would they be taking a unit a day? Would they be taking 10 units a day?
I don't know. I mean, there's no medically recommended dose, so I actually don't know what dose like President Trump was taking. I don't know the answer to that.
Okay. There are questions piling up, but I do want to ask, because we talked, today's title was, what tests are in the field? And perhaps you can touch upon which one, because you said there are 1,000. So if you could spend a second on each of the 1,000, that'll just take us a few hours. Maybe the top 3 that you are particularly excited about, promising, that we should be looking for?
Yeah, so I think, we talked a little bit, there's the RT-PCR test that tests for the presence of the virus. And those are pretty good. They're easier to make, a lot easier to make than an antibody test. An antibody, you have to express the protein in the right conformation to be able to have the antibodies bind to it. With the RT-PCR,
places where we look to track that study, is it a study? Is it 100 studies doing the same thing? Is there a site that says, here's the series of letters you
the Johns Hopkins site does pretty well with the Johns Hopkins. They track the nucleic acid tests. Okay. You can Google Johns Hopkins COVID, and they have a site that that summarizes the positive tests. Then the antibody tests are much more complicated, and the FDA allowed a lot of emergency use authorizations early for these tests, and there are a lot of bad ones out there. So I think the rapid tests I wouldn't trust at this point because we have a low prevalence setting and that false positive issue. You have to have almost perfect specificity. So the best I've seen is the ortho test. The Abbott test is also quite good. Those are the two that I've seen.
That's the one they just came out with last week saying, you know, expect a high false positive rate, the Abbott tests.
Well, yeah, so there were two. Abbott had a nucleic acid test that was not sensitive. So when you put the swab into viral transport medium, something bad was happening and they were getting 15% false negatives. So that's bad. And then their antibody test, it's 99.6% specific. So if you're in a low prevalence setting, as I said, in San Francisco, 3 of the 4 positives were false positives because we have a very low prevalence here. If you go to somewhere like New York, we have 10 to 40% positive, depending on the neighborhood, then that level of specificity is just fine because most of your positives are going to be true positives. So that's where you have to look at the test where you're applying it. So antibody tests are more useful at the population level than on the individual level at this point in the epidemic. And you know, why would you test an individual like yourself? You're going to go to the blood bank and get a test, which is very good. If you're donating blood, you're going to get this test result back as an incentive to come in and donate. That test is very specific, and it probably means you're positive if it's positive. But does that mean you're protected from the virus? We don't know. How long will you be protected? How long will these antibodies last? Well, we don't know that either because the disease hasn't been around that long. We can look back at SARS-1 that was circulating in 2003, and we know that the antibody does tend to fade over a year or two. The T-cell responses stick around longer, so hopefully you would have some memory, immune memory from your T-cells, and if you got infected again, they would rapidly come back up and your B-cells would come back up. So that's what we would hope, but we don't know that yet. So if I were, and we'll talk about this next week probably, if I were thinking about testing, I would rely more on testing for the virus than testing for the antibodies, because you really want to know who's infected in your cohort and weed them out and quarantine them.
I try to keep these to an hour. Maybe we'll go over a little bit, and I do record them. The questions I'm getting are complex enough that we would. So I think what I'll do, we have the great luxury of having Philip back next week. Maybe we'll come prepared to answer an awful lot of these. Let's see if I can't get 1 or 2 of these in. Kimberly asks, what's your opinion of saliva-based testing? What data exists besides the Rutgers data that indicates this may or may not be available specimen typing for testing?
Yeah, no, I think so. In our longitudinal study that we're doing, we're planning to do saliva testing because it's not quite as good as sticking the swab to the back of your brain, but it's pretty good. I think it's maybe 90% as good. The viral, there have been a few studies beyond the Rutgers studies, and so if you take the same person and do a nasopharyngeal swab and do a saliva sample, and I think they did an oropharyngeal swab, they found that the highest viral load really was that nasopharyngeal swab that went to the back of the nasopharynx. But saliva was pretty good. You know, maybe it was 5-10-fold lower viral load, and you get pretty high viral load in the upper airway. That's how you're transmitting it around. So you would miss some with the saliva, but if it's so much easier to collect, you could do these at home. You know, there are saliva collection cups. They're kind of a funnel-shaped thing with a tube at the bottom. So you spit into this bit of a funnel, and it goes down into a tube, and then you can cap it and send it in for testing. I think that's going to be the future. So if you're thinking about where to go now, it's a matter of convincing the FDA.
Well,
they've been
and the public. I mean, so my line of work is marketing, communication, and strategy, and from a communication standpoint, I'm confused as hell, and I'd say I'm probably paying more attention to some of the details than the average person.
Sure.
So, you know, for example, when I said, oh, I heard Abbott had a false positive thing, you said, oh, well, there are 2 Abbotts. And I was like, oh, I didn't even realize that. So just like the name Abbott became kind of diluted to me as a reliable place to go for testing. I just think that it's going to be a communication nightmare to get, and not to mention that it depends on what network you're watching. It's just so dispiriting. I can't even, okay. Just smiled through that.
I don't watch TV.
Well, that's one answer. Just as we were going on about Mad Cow, Jan recovered from cancer 28 years ago. She wants to know anything on cancer patients donating blood.
Donating blood. So I think the type of cancer determines if you can be a blood donor or not. You would have to, there's an elaborate questionnaire that is given to any prospective blood donor.
She's taken it and was told no.
Okay, well then that would be a no.
Yeah.
There are some cancers that you're disqualified for life.
Hodgkin's lymphoma.
Okay, that's a liquid cancer, that can move around, so that's probably why.
So forever no.
As far as I know, but I work at a blood bank, but I'm not a medical director here, so I don't know that form like the back of my hand.
She says rats.
Sorry.
Yeah. Yakov asked 2 questions. Why can't you draw blood or other cells, infect them in vitro, wait until the antibodies respond and multiply, and then put it all back into the bloodstream?
Well, so that doesn't work because I mentioned the lymph node is where it happens, and that's in a person. So your body is an amazing factory for all sorts of things, and antibodies are one of them. But there are things that we're doing that can be done in the lab. So the first thing we're doing is giving convalescent plasma. So we get people who've recovered, we come take the liquid part of their blood, the plasma, we collect that, and we can give that as a dose because it's loaded with antibodies. The next level of refinement are the plasma fractionators. So they collect plasma from 1,000 people, mix it all together, pull the antibodies out. So rather than giving the whole liquid part of the blood, you're giving a concentrated antibody dose from people who have that COVID antibodies. So that's the next level of refinement. Then the next level after that, and one of the things we're working with, so we've identified people who have strong antibody responses. We can then take their cells and have them stick to a SARS-CoV-2 protein, the spike protein, and the cells that stick are the ones that produce antibodies that recognize that spike protein, and then we can clone those cells. So you can get single copies of those cells, you get it alone in a dish, and you stimulate it and get it happy, and it will make billions of copies of itself, and then that is an antibody factory that's producing what we call a monoclonal antibody. So that's a single antibody rather than taking all the antibodies in the body, a portion of which are SARS-CoV-2 specific. You can have a monoclonal antibody, and people are all over the world trying to develop these monoclonals that then you can grow in a lab and you can give them as a therapeutic. So people are working on that too.
Okay. With just a few minutes to spare, I'm going to ask you 2 questions, and then I'm going to say a thing or 2. Yakov still asks, for simplicity, he asks, how bullish are you on CRISPR-based tests? Real short answer, bullish, not bullish.
Bullish.
Good.
I mean, they're, at least in concept, they still need to be tested and all that.
I'll remind everyone, Philip is back next week. And before the call started, I told you that, when I do any call, sometimes as few as 1 in 6 people show up.
Right.
2 in 5 people show up, and they stay the whole time. So there's demand for your knowledge, and I really appreciate what has got to be a very tight schedule that you made time for us. Fred asks, are blood samples from donors stored and archived? Could you basically retroactively go back into the bank and test against the archived blood? Yeah,
really good question. So in the U.S., in some countries, you have to keep a sample of every blood donor for 2 years, like in Vietnam. So they have 2 years of, and every month they throw out 2 years ago that month's samples. So they have archives going way back. In the U.S., we don't. We have so much more blood collected. We do make archives for research studies, so we do have archived blood. And in fact, we have plasma collected before the epidemic, and we're giving that as a control for these convalescent plasma studies. I think the best, I think you're trying to get at the question of when did COVID arrive in the US. So a couple of things we did. So one, I mentioned that I thought I got infected in January, and we had the earliest antibody tests here in our lab. So I went around, and there were some clusters of respiratory virus in Sacramento and Davis and the South Bay that had good stories for COVID in January or December. And so we looked at antibodies, and in none of those clusters did we find anything. And then in a more systematic way, there's a guy who runs the virology reference lab at University of Washington in Seattle. And so he's done some really nice work. Sorry?
What's that person's name?
Oh, I can't remember. He has a Twitter account.
Okay.
I'll find it and send it to you. He's really great. So they test, thousands of samples a year for respiratory specimens, like that swab I talked about, and they'll test for all sorts of viruses. So they had in their repositories, they were able to go back to December and January because, February is when things really got hot in Seattle. And he went back and looked at all these respiratory pathogens from December and January and added on SARS-CoV-2 testing because they developed a test by that point, and none of those samples tested positive. So if there was any community transmission early, it was really small, and it was not coming to the attention. It wasn't really presenting to the medical professionals.
Before we close, I invited my friend Peter. I opened his mic. This may be a bit tangential, but he's working on something that he wanted to bring to the attention of the group. Peter?
Hi, greetings all. My name is Peter Belshaw, and I am the CEO of ImmuneSciences.ca, it's a Canadian company working on diagnostics and therapeutics and some vaccine-related technologies. We are an intellectual property generation company, and we're looking for licensing partners. But I'm also forming an institute, which is going to be based kind of on academic licensing models, sort of a 30-30-30 to inventors institution of where the invention came from, and 30% to the parent institute. I'm also available as a fee-for-service research arm, potentially, if the research projects are aligned with our expertise. And our scientific advisory board is currently at about 12 of the top scientists in North America, but mostly about half US and half Canadian. So if people would like to reach out to me to find out about potential research collaborations, I can be reached at peter.belshaw, BELSHAW, at immunesciences.ca.
Thank you, Peter. And you're in our Slack channel, so you can go ahead and share that information, please, in one of the channels as well.
I will do that later this evening.
Thank you very much. Dr. Philip Norris.
Peter, where are you based? Just out of curiosity.
I'm just on the western side of the Greater Toronto area.
Okay, so I'm heading up to Muskoka in a couple of weeks.
I have a cottage in Muskoka. We should connect.
Yeah.
Look at that. See, I'm bringing people together. You get to stay at Peter's place for free in the cottage.
I love it.
And we said I wouldn't compensate you for this hour, but there you go. So it's all coming together. Dr. Norris, I'm exceedingly grateful. I know the audience is too. And we'll see you next week. Go ahead, please.
Okay, that sounds great.
If you can't get approval to share those slides with me, I'll share it more broadly with them.
Okay, great. Yeah, I think I probably can. I just have to talk to the two scientists.
And one more thing, folks. I wrote an email to all of you I did it Friday, I resent it yesterday. It's getting hard for me to discern how much continued interest there is in this initiative of us having a weekly call and the group and everything. I think we're getting a little coronavirus tired, and that it's 24 hours a day. We hear about this, think about this, and at what point is enough too much? So I'm looking for your feedback to please write me and let me know how we can best channel the energy of the group Maybe we do it less frequently. Maybe we do it in other ways. Maybe this evolves. So please do let me know how you're feeling about these calls and our initiative. I'll see you next week, Philip. Thank you, everyone.
Bye, everyone.
Bye.